Welcome to the Evolution of Immunity module. In this talk, we're going to look at the different defenses that vertebrate animals employ. We will first begin looking at the different mechanisms that have been evolved by prokaryotes, followed by protists, and then invertebrates to defend themselves from other organisms. We will then learn about the mechanisms that vertebrate animals have evolved to defend themselves. The immune system in higher organisms provides protection against a wide variety of pathogens that can include viruses, bacteria, fungi, protists, and helminths. They're able to do so by employing a wide variety of strategies, which includes the production of many types of antimicrobial compounds, the synthesis of different proteins that have the ability to recognize pathogens, and the evolution of mechanisms to make variations of the same type of protein that enables the recognition and destruction or elimination of different types of pathogens. The immune system of higher organisms also has an elaborate mechanism to retain the memory of pathogens. Such a highly complex system is a product of millions of years of evolution. In order to understand how such a complex system has evolved in higher organisms, let us first look at the different defenses that lower organisms or simpler organisms like prokaryotes and protists have evolved. Prokaryotes are the earliest living organisms on Earth and belong to the kingdom of either eubacteria or archaebacteria. They compete with other organisms for space and nutrients and hence have evolved different mechanisms to protect themselves. These include producing substances to prevent the growth of other organisms, and they're able to protect themselves from non-living entities like viruses by preventing viral adsorption and entry, employing enzymes like restriction endonucleases to degrade viral nucleic acids, disrupt the viral life cycle, and evolve a complex mechanism, which is the CRISPR-Cas system that maintains memory of the virus and also protects them from viruses. Protists are eukaryotes that evolved from archaea, and they belong to the kingdom of Protista, which is the most diverse kingdom of life. They also have evolved mechanisms of protection, and some of them are evolutionarily conserved, like the production of toxic substances. They also have specialized organelles like lysosomes for degradation of different types of microorganisms that they may have taken up. They also produce other types of membrane-bound vesicles like extrasomes, which can provide mechanical as well as chemical defenses. They do use a mechanism of RNA interference to protect themselves against viruses, and this mechanism has some similarities with the CRISPR-Cas system. Some protists evolved into organisms that belong to the kingdom Animalia. The first animals that evolved were the invertebrates, and they also employ different protective strategies. Some of these protective strategies are evolutionarily conserved and include the synthesis of toxic substances and using RNA interference to protect themselves from viruses. They also were able to evolve other strategies like producing pattern recognition receptors that are able to recognize different components on microorganisms. They were also able to make cells that could move around throughout the body to protect the organism from different types of pathogens. Additionally, the invertebrates evolved genes that were able to give rise to many different protein isoforms to aid in the elimination of different types of pathogens. An example of one such protein is the dsCAM protein. Some invertebrates evolved to form organisms with a vertebral column, which are called as vertebrates. All vertebrates belong to the phylum of Chordata and the subphylum of Vertebrata. There are different classes of vertebrates, which include the fish. These are the ones that evolved first. Amphibians then evolved from fish, and then some amphibians were able to evolve into reptiles. Reptiles then gave rise to birds, and some other reptiles were able to give rise to mammals. Thus, there are different types of classes of vertebrates. The first vertebrates that evolved were fish. Fish can be grouped into three main classes, which are the agnatha, and these are fish that do not have jaws, the chondrichthyes, which are fish that do have jaws but have cartilage instead of true bone, and then the osteichthyes, which have true bone and are jawed. The jawless vertebrates, or the jawless fish, were the ones that evolved first. There were many types of jawless vertebrates in the past, but right now there are only two main groups, and they are the hagfish and the lampreys. Vertebrates are much more complex than other organisms like invertebrates or protists, and thus they have to have multiple strategies to protect themselves. Many strategies are similar to what is observed in invertebrates, which includes the synthesis of toxic substances and the production of pattern recognition receptors. Vertebrates also have cells that can move throughout the body to protect the organism, but they have more types of cells for this purpose compared to invertebrates. Vertebrates also show the presence of RNA interference strategies. Jawless vertebrates have also evolved genes that can give rise to many different variations of the same protein to aid in the elimination of pathogens. Now, this strategy is also observed in invertebrates. However, the mechanism that jawless vertebrates employ is different than what is observed in invertebrates. These proteins are normally made in the lymphoid cells that are a type of immune cell present in the jawless vertebrates. Jawless vertebrates have lymphoid cells, which are a type of immune cell, and these lymphoid cells express variations of a protein called as the variable lymphocyte receptor. The variable lymphocyte receptor has many leucine-rich repeats, or LLRs, as we can observe in the domain structure of the protein. The variable lymphocyte receptors can bind to different parts of infectious agents, and they're encoded by the VLR genes. 3 VLR genes have been identified to date and have been named as VLR A, B, and C. When we look at the germline VLR genes, what can be observed is that the DNA sequences are quite different than the corresponding sequences that are needed to give rise to a functional VLR protein. The functional VLR protein is going to be made in the part that's shown in the blue box, but as we can see, a lot of parts of the VLR protein are actually missing in this region. What scientists have found is that on the 3' and 5' end of this region, there are DNA sequences called as LLR cassettes. So at the 5' end, we have the 5' LRR cassettes, and at the 3' end, there are the 3' LRR cassettes. There are more than 350 LRR cassettes, and so it seems like the germline VLR genes are like an unsolved jigsaw puzzle with pieces all over the place. An example of some of these cassettes is here. We can see 3 types of one of the cassette, and in the functional protein, only one of these cassettes can be chosen. Similarly, here we can see 2 other types of cassettes. Here's another type of cassette, and thus all these different types of cassettes have to be rearranged in order to form a functional VLR gene. The incorporation of different LLR cassettes in the part of the VLR gene that will then ultimately give rise to a functional VLR protein occurs through the process of DNA recombination. and thus we don't see alternative splicing as a mechanism to generate the VLRs. The somatic recombination occurs in a stepwise manner where cassettes are chosen and then incorporated into the region of the VLR gene that will be transcribed and then translated. So by the stepwise addition of the cassettes, the functional VLR gene is then formed. Now these different cassettes that are chosen during recombination have to occur in such a way that only certain cassettes are chosen. This is done in a random manner, and hence this is a way of obtaining diversity because we are able to form different types of VLR proteins due to the random picking and choosing of different cassettes. This process can result to different VLRs, and it has been estimated that we can have up to 10 to the 14 different types of VLRs being produced. What should be noted is that even though we can have a variety of VLRs being produced where there are small changes in their amino acid sequences, each lymphoid cell can express only one type of VLR. Thus, different lymphoid cells can express different variations of the VLRs, but each lymphoid cell will express only one variation of the VLR and not multiple variations. Now, upon infection, the VLRs are shown to bind to pathogenic components. And when this occurs, the outcome can be in the form of secretion of some types of VLRs, which results in the effective elimination of pathogens. So as we can see in the figure, lymphoid cells that express the VLRB gene are able to make VLRs that can be secreted when a pathogen or an antigen is present in the environment. Other types of lymphoid cells that express other types of VLRs, like in this example, lymphoid cells expressing VLR-A, can produce molecules that are soluble in nature, and these molecules aid in the clearance of pathogens by activating other types of cells. This is an example of the adaptive immune response that is observed in jawless vertebrates. Some jawless vertebrates gave rise to the jawed vertebrates, and jawed vertebrates include most fish, all amphibians, reptiles, birds, and mammals. The jawed vertebrates show most strategies that are employed by jawless vertebrates in order to protect themselves from microorganisms, and these include the production of toxic substances as well as pattern recognition receptors. Jawed vertebrates also have an entire array of cells that can move throughout the body to protect the organism, and these are the immune cells. They also show RNA interference strategies, and thus we are able to see many evolutionarily conserved strategies. Jawed vertebrates make lymphoid cells, which are types of immune cells that express variations of a protein that contains the immunoglobulin domain, which is also abbreviated as the Ig domain. These lymphocytes come in two flavors. We have the B lymphocytes that make the B cell receptor and the T lymphocytes that make the T cell receptor. Here is a picture of the T cell receptor, and we can see it is made up of two polypeptides. When we look at one of the polypeptides and look at the gene structure for that polypeptide, then what we notice is that there are these gene segments. Thus, the B cell receptor and T cell receptor genes do have these gene segments, and these gene segments have to be rearranged in order to form the functional protein. The gene segments can be rearranged through the process of DNA recombination. In this process, one V gene segment will be joined to one randomly chosen J gene segment. This process is similar to what we observe in the variable lymphocyte receptors that are found in invertebrates. However, the mechanism by which this process occurs is different because it uses a different set of proteins. In the case of B cell and T cell receptors, the protein that mediates this DNA recombination event is the RAG recombinase. Now, once the V and J gene segments are joined together, then the DNA can be transcribed. And the RNA that is then formed is spliced, and finally we are able to form the polypeptides that form either the B cell receptor or the T cell receptor. It should be noted that each B or T lymphocyte will express only one type of B cell receptor or T cell receptor. Even though individual B or T lymphocytes will express either one type of B cell receptor or one type of T cell receptor respectively, different lymphocytes are able to express different variations of the B cell receptors and T cell receptors. The B lymphocytes and the T lymphocytes are able to form the adaptive immune response of jawed vertebrates. When we compare the VLRs and the B cell or T cell receptors, what we can observe is that in both scenarios, the genes cannot make a functional protein. In the case of the VLR gene, there are many leucine-rich repeat cassettes that have to be rearranged to give rise to a functional VLR gene. Similarly, in the case of jawed vertebrates, the BCRs and TCR genes are made up of gene segments that have to be rearranged to give rise to functional genes that can code for the appropriate polypeptides. In the case of the VLR genes, DNA recombination has to occur in order to form the functional VLR gene. This is true in the case of the BCR and TCR genes as well. However, the mechanism by which recombination occurs utilizes different players, namely the RAG recombinase complex. Once the VLR gene undergoes its recombination and is able to code for a functional VLR protein, the VLR proteins are then membrane bound. However, when there is a pathogen present or a pathogen component like an antigen present, then the cells are able to secrete the VLRs into the environment. In the case of the B lymphocytes, we're able to see that the B cell receptor that is present on the cell membrane is also secreted, and these B cell receptors can also be called as immunoglobulins or antibodies, and this secretion occurs in response to a pathogen or a pathogen component. Thus, we can see similarities and differences between the VLRs and the BCRs and TCRs. Furthermore, scientists have found that the B lymphocytes that are found in the jawed vertebrates are actually similar to the lymphoid cells that express the VLRB genes in the jawless vertebrates. Similarly, one subset of T lymphocytes that are formed in the jawed vertebrates are similar to the lymphoid cells that express the VLRA gene in the jawless vertebrates. Finally, the second subset of T cells that is found in jawed vertebrates is similar to the lymphoid cells that express the VLRC gene in the case of the jawless vertebrates. With this, we come to the end of our talk, where we learned about the different mechanisms that prokaryotes, protists, invertebrates, and vertebrate animals have evolved to defend themselves.